Hello everyone, Microbehunter here. Welcome again to another Saturday microscopy live stream. Happy that you could join in again. And over the next hour 15 minutes maybe, I would like to unpack and unbox this slide box which I just bought. It's still packed originally, so I did not open it. There's still a plastic wrapping around it, as you can see here. And I would like to unpack it. I really have no idea what to expect. I bought this over Amazon. It was not so expensive. And what I would like to do is I would like to unbox it. Of course, I would also, of course, then put some of the slides under the microscope. There are, as according to the description, 100 different slides on here. Before I unpack it, we're going to have, of course, also a look at the box on the outside first. I see that there are already people joining in from all over the world. So there is hello from everyone from Northern Germany. The sound is okay. Thank you very much. You can see that I'm not wearing my headset anymore. I do have a microphone next to me. I hope that the sound is a little bit better this way. Hello from England, from the Netherlands, from Austria. I'm also from Montreal. I love it. Microscopy is enjoyed all around the world, and I'm happy that you could join in. If you're new to this live stream, just very quickly the guidelines or the rules. I will be interrupting the live stream. I'll be interrupting myself, the talk that I'm giving, occasionally to read the comments that you have posted in the chat. And I'm going to try to answer some of the questions, and as long as they are somehow microscopy related, I will try to answer them, even if they are not directly related to the live stream, but as long as they have some kind of a connection to hobby microscopy or microscopy, then I'll try to answer those. If you have specific questions for me, please put an @ at Microbehunter or at Oliver. So that I can more quickly find those comments that are relevant for me, because sometimes, the viewers are also going to, communicate with each other, and it's easier for me to find it if you actually make a little @ okay? So from Montreal, from Belgium, hello from Switzerland, from Japan, okay, from Rhode Island, the USA. Okay, here we go, and we're going to do the following. I'm going to start right away, and we're going to have a look here at the slide box. So first of all, as I mentioned, I bought the slide box from Amazon. I did put an affiliate link also into the description in case you're interested, but don't order anything yet. We're going to have a look first, okay? And the box here is surprisingly large. Okay, it's fairly thick. It actually doesn't say anywhere over here how many slides there are included, but it's a box for 100 slides. There is a sticker here on the side that actually mentions that it says here 100p over here. Maybe that means 100. I don't know. So it is, also made in China, the whole thing. Doesn't mean that it's this kind of explains maybe why it was relatively cheap. Because there are also companies, I know of a company in Germany who are selling prepared microscope slides for educational and for training use. And these slides are made in Germany. They're very high quality slides, but of course they're also more expensive. So let's have a look here at the box first. There are 4 little pictures on here. Insect, that's correctly labeled. Okay, that is an animal tissue, also correctly labeled. Looks like a little bit like the intestine. Plant cells also correctly labeled. This here says tissue. Of course it's tissue, everything's tissue. Okay, so they couldn't think of a better thing here. More interesting is the thing that's written over here. Yeah, of course, that's the standard slide size. Yeah, that's clear. The thickness is completely irrelevant for a slide, of course. Pre-cleaned slides is also irrelevant. They better be pre-cleaned, otherwise they're not useful. Frosted rounded edges. Now, what they actually mean is, and that is interesting here, is that the corners of the slide here, they are cut. Okay, so that means there are no sharp corners, but that's actually not a frosted edge. This is actually frosting. If the slide itself has a frosting so that you can write on it. And highly transparent glass, of course, it's going to be highly transparent, otherwise you're not going to see anything. Okay, so the question is, where do I normally buy these slides? Generally, this slide box I bought on Amazon. There is they sell it on Amazon.com and also the German Amazon, so they sell sell it. Yeah, I guess they're manufactured somewhere and then they're basically just imported and sold. It's not specifically a recommendation, or I'm not recommending it or not recommending it. It's just they're fairly cheap compared to other prepared slides. But again, I do not know how good the quality is. Okay. There is no more motion. The problem was local. Okay, good. Okay, I've got a comment that somehow the video froze a little bit. If the video really completely interrupts, okay, if the video really, I'm going to turn down the sound a little bit. If the video really completely interrupts for whatever reason, which happened a few weeks ago, and if I'm not able to restart it, I will have to create a new video link, okay, a new live stream link. So please do check then in my YouTube channel. There should then be, after a few minutes, a new link appearing, and then you have to click on that and you have to restart the whole thing. Yeah. So really, I have no idea what to expect. We're basically all on the same level, and we're just going to start unpacking it. So I'm going to I've got some scissors here. I'm going to just cut open, make a little cut here, and I'm going to remove the plastic cover. Okay. And unfortunately, as so often I think the description is not very detailed on Amazon. So this is a little bit unfortunate. I would have liked to see a little bit more information on what is actually included. Okay, so that's number one. Plastic goes away. We carefully try to open it here. I don't want to damage the cardboard. This is still a challenge. Okay, yeah, here we go. Okay. Here we go. Okay. Now I know why the box is so thick. Okay, it's empty. Let's put this away somewhere. Because it has been quite well padded. Okay, here we go. Yeah, plant, insect, human tissue. Okay, fine. Microorganisms. These are not microorganisms. These are also tissue cross sections. It doesn't matter. I'll not be too. Too picky about that. That actually doesn't look like human tissue. It looks like a little bit more like spores or something. I don't know. Doesn't matter. There is a brand over here. Max Lapter. Okay, never heard of that brand before. Let's see if I can open it somehow. How do I open this? Okay, always be careful. Here we go. Oh yeah, looks quite nice. Is there a label? Very nice. It's also properly labeled. Young root. Okay, Z root, Z maize, root of. Oh, I can take this out. So maybe I can actually show this to you. There is a focusing possibility. Does this focus? Yeah. So that's basically what's included here. Rapeseed flowers, lower epidermis of orange peel, sunflower stem. That's actually one of my favorite ones. Is always the sunflower stem, pine stem, of course. CS means a cross section. WM means whole mount. TS means, I don't know, maybe transversal section. And LS means longitudinal section. So basically they tell you a little bit of how this was cut. Okay. Honeybee wings, here the insects, pollen carrying leg of a honeybee, human blood smear, cockroach mouth parts, female mosquito mouth parts. Okay, I mean it looks Hirudo, I don't know what that is. Some worms, would be interesting to see which ones. Basically it kind of looks. I'm happy that they actually have included this here as well, obviously. Let's try a few of those. I don't know. I'm just gonna start here at the top. Young root of what does it say here? I've got a broad bean. A root of a bean. Okay. Yes, that's correctly labeled. Okay, so that's good. So what I'm going to do is now the following. I'm going to, I have to refocus here. My camera has this autofocus function here, and now we're back in focus. Maybe I'm also going to turn on a little bit the light here. No, that's not needed. So what I'll be doing is I'll be putting those under the microscope, and I want to actually check for two things. Number one, I would like to check a little bit. Is there any dust and dirt on the slide? And I'm not so much concerned about the dust and dirt here on the side, but rather have beneath the cover glass. And the second thing that I would like to check a little bit is how thick are the cross sections, because I did once buy some slides and they were otherwise okay, but they were cut so thin that the specimen was kind of very pale. It just didn't look very nice. Okay, not a problem as such. I mean, it just we're cutting it thin, but I kind of would have preferred it a little bit to have the sections made a little bit thicker. Okay, so this is a little bit the thing that I would like to check now here. And there are already a couple of issues here. I also have problems with video lagging. I was not sure if it was only my computer here. Okay. Yeah, so maybe there seems to be some internet issue. And there's a question. Can you explain a little bit the difference between all of those LSCS, etc.? Okay. You know, that's actually a good point. Okay, so I'm going to put this back here, and I'm going to use I'm going to use my computer mouse here as an example. Okay, this is the specimen, in other words, and you see that the specimen has a certain extension, and if I take the whole specimen and mount it, then it's called a whole mount. It's labeled WM. If I basically cut the specimen like this across, okay, and put that slice on the microscope slide, and it's called a cross section. And if it's cut like this, it's a longitudinal section. So this is especially then relevant if you have a certain tissue that is different in whichever way you look at it. So for example, muscle fibers. Muscle fibers are long fibers, and depending on how you cut it, they look quite different. Yeah, I'll give you an example again. I don't know. Do I have some other? Yeah. I mean, you get the idea. If you cut something, depending on which direction you cut it, this kind of tells you a little bit, of how it was processed. Yeah. So there are some video issues here. I've refreshed the video. As long as I stay in the last second of its live, it's okay. But if I go into the past, it's quite laggy. Okay, if you go into the past, it might take YouTube also a couple of seconds to process the video. So this is the thing. Is there such a thing as prepared specimens for stereo microscopy? Interesting question. Of course, you can also look at those slides under the stereo microscope. However, the magnification might not be quite as high. We can have a look as well. But generally, stereo microscopes, what you do is I mean, what you can try is that I've seen some people actually putting some specimens into resin, which is epoxy resin, and then essentially you have also prepared it. Yeah. So what I'm going to do now is I'm going to switch over to the microscope view. Yes. Okay. You can also see the stage, and I'm going to flip out you see, I've got this condenser that can be flipped out. Okay. I'm also going to remove now the DIC. So this is now the young root of a broad bean. So this is going to be interesting now. I'm not going to look through the microscope because, I do see a little bit here. This is where there's always a little bit of dirt on the outside, which is, I guess, fine. I'm more concerned about the dirt that's in here. Here it is. And let me close the condenser a little bit. Yep. I can already see that it is cut quite thin. Okay, so that's the cross section of a root. I go up, you know what, I put on DIC just for the fun of it. Okay, I have to refocus. Yeah, we can see the individual cells here. Yeah, so that's a little bit how it looks like. The specimen seems to be stained as well, of course. And we again go up one higher magnification. Yeah, we can see the stained nuclei. Those dark dots that you see, these are sometimes stained nuclei. What appears to be. And we're going to kind of 40x. I can, of course, adjust the background here as well. Okay, let's have a look here. let's go a little bit outside of the root. There's a little bit of dust here, but I have seen worse slides, honestly. Look at this little thing that you see over here. That's a side root appearing. Yeah. My first impression is, at least of this specimen, here you see that over here, the cells, they have a dark purple dot. That is the nucleus, so this slide is stained. You see that over here, most of the cells do not have a nucleus, and the reason is because it was cut so thin that the cell organelles kind of were removed. And the cells over here seem to be much smaller, and therefore they were not cut open, and therefore you still see the cell's contents. Okay. Yeah. So yeah, I think it's usable. It's usable. You can also see over here that some of the cell walls are thicker than others. Yeah. Also stained differently, so maybe they use differential staining as well. Yeah. So it's possible to use multiple stains. So you first stain it with one substance, and then you add another one as well. Okay. So generally, the first one is okay. I still have 99 to go. Okay, this basically means I'm not going to finish everything here. So this means what I'll be doing is I'll be going through the slide box as far as it can go, and maybe what I'm going to do is over the next week, I'm going to probably have a look in more detail myself, and then maybe next week I'm going to show you some more. So that is now ZMAs. That is now the root tip of, that's very nice. It says here the longitudinal section. That is the root tip of corn. Okay. But by the way, corn. Look what I got here. I've been growing some popcorn myself. Okay. Here, okay. On tissue paper, it's because I was actually also trying to do a staining reaction with that. And here, this is now a longitudinal section. Here you can see, and I don't know if you're able to see this here. It's actually lengthwise. So we're going to put this also under the microscope here. Okay, I'm now at 40 times finding it. I'm lucky that I already got this here. Okay, yeah. So let's try to find the actual tip. Here is the tip. Okay, it always goes a little bit out of focus. But you can see that here, the tip is the so-called the apical meristem. This is where cell divisions actually happen most. And so there are lots of cells here. And this is the growing part. And the further back you go, the longer the cells become, because the cell starts to grow in size, and that's why they are much longer here. Yeah, I don't know. I won't have the time to do that now. No, I lost it. But maybe because we can see the nuclei as well, maybe it is even possible if we look very carefully to see some cell division stages. Okay, that would be kind of very nice. Okay, 60 times now. It's becoming darker and darker. Let's try to so I've removed now DIC. Okay, yeah. Yeah, I think it's quite usable. All of those little tiny circles that you see in the cells, that's of course the nucleus. Yeah, so that is the root tip of corn. And you know what I'm going to do now is I'm going to go a little bit back to the chat again. Okay. Reading. At Microbehunter, do you use more than one stain to get multicolored different parts? How do we? This depends. How do you actually use multiple stains? It depends very much on the staining protocol that you can use. In most cases, you only use one stain, but there are for certain stains you can actually use multiple stains. Yeah, hematoxylin and eosin, for example. So there are sometimes you can actually use different stains as well. Yeah. Sometimes you prepare it with one stain and then you stain it with a second one. Is Amscope a good brand for microscopes? Because I'm thinking of getting a beginner one there. Look, it depends more on the microscope than on the brand. And what is good and what not good is really also to a certain extent a question of personal taste. Yeah? Methylene blue and iodine. Okay. What am I watching? Some people joined in. So basically what I've been doing is I've been unboxing a slide box, which I recently bought. And I'm now looking at the slides. And if you joined in late and if you want to see the beginning of the live stream, then don't worry. After the live stream has ended, the video is of course still available. So the next one over here is root of Gossypian. Oh gosh, difficult to pronounce. Also root cross sections. So they seem to have sorted the whole thing now based on plants. And let's try to here it is. Okay. I generally like plant cross sections because they make beautiful patterns, a little bit like modern arts. And yeah, this one seems to be, it's also quite nice. See the different sizes of the different cells. Yeah. Okay, here we go. Again, here on the side, you see a couple of cells that have broken off due to the when they were preparing the slide. But otherwise, there is not so much dirt on here. There's a little bit of, of course, there's always a little bit of stuff here, but it's not so much compared to some other slides that I've seen. So I think, for the price that I paid, I think it's quite okay. So I'm going to go through this here. There are a lot of plants here. The sunflower stem, number 14, I'm going to jump ahead because I do have in my other slide box a favorite one, and that's also sunflower. Okay, here that's a little bit difficult now to see where is 14. Just a second, I need to change over to the desk view, otherwise you don't know what I'm doing. Okay, here I have to. There's a number on the bottom here. Sunflower stem. Okay, just by looking at it, it's relatively pale, and this actually means it's cut quite thin. So let's go again. You have to change back to the microscope view again. Never forget doing that, otherwise, So here we go. Okay, that's again a sunflower. So what we see here is the typical arrangement of a so-called dicot plant. So-called sunflowers are so-called dicots, and this basically means that all of those unfortunately I don't have the arrow right now because I set up the computer and I forgot to include the arrow. Unfortunately, but what you see over here right in the center, this cluster of cells, that is a so-called vascular bundle. It's right in the middle here. You see that there seems to be some kind of an aggregation of cells. This is where water is transported up the stem, and where also nutrients are transported down. And those circles that you see, these are the cell walls. And back in the day when microscopes were invented, and when they made their first observations of plants and animals under the microscope, in plants they could see those cells. And at that time, they were very fascinated with the cell wall because it's the most obvious thing to see. And at that time, scientists were even kind of wondering, because they didn't know, they were wondering, could it be that this the cell wall is the living part of the cell, and the central part is just empty space, like a sponge maybe? So this is something that they were not even sure of. Now, of course, we know that the inside of the cell is actually the living part, and the cell wall is a structural component, which is strictly speaking outside of the cell, because the cell is bounded by the so-called the cell membrane, and then you have the cell wall, which we see here on the outside. So we know that now, but at that time, the scientists actually, they were kind of wondering, could it be that, back in the 19th century, could it be that the cell wall is actually the living part? And these are just empty spaces between the cell wall. Okay, so they were not sure about that. So there seems to be a video lagging issue. So what I'm not able to do that now, I think. But but let me check something. Stream. Unfortunately, I cannot do that now, but what I will do for the future is I will go down a little bit with a bit rate so that there is it's going to be hopefully a little bit more smooth. Okay, so so this is another plant one, but I'm just gonna go through a few more that we have here. Desk view. So the sunflower, you see those plants do look quite similar. Rice stem. Okay, I hope that we're able to see that the vascular bundles are a little bit scattered throughout the stem. That's not a stem of rice. It's quite a large cross section that we have here. So, here we go again. This does not look so nice here. Here we go. That's a little bit better. Okay. Okay, because it's hollow. Okay. Yeah, this one is again here. We are able to see the different cells. It is not. You see that there are some holes in the specimen. Okay, so that's basically due to the cutting process. This has caused the tissue to rip and tear a little bit. This is sometimes the case, like over here. This is sometimes the case when the specimen is indeed a little bit too thin. I wonder what all of those dots are. Yeah, what are they? I don't know. Okay. Yeah. But they do seem to be part of the specimen and not on the cover glass. Okay. So here we go. Here again a vascular bundle. What we see are those large cells. Yeah, that seems to they're next to each other. That seems to be the xylem, which transports water up the plant. Okay. So let's have a look here. Cross section. Yes, somebody actually. Thank you very much. CS cross section. Okay. Do you added a few more things here? SM smear, ST stained, SQ squashed preparation, transverse section. Thank you very much for that. Okay. Question for me. Do you think that the video is lagging? I don't know. Okay. Many people have actually said that the video seems to be lagging. What I will do now is the following. At the same time, I'm also recording my stream. Okay. On my computer. So I will stop recording now. And now I'm only streaming. I don't know if this actually solved the problem or not. Okay. So this is rice. Let's move it again, remove it again, and have another look. Yeah, for those of you who joined in a little bit later, basically what I did is I bought myself a slide box, okay, which looks like this, and I opened it, and we're just looking at a few of these specimens here. And for those of you, if you feel that it's worth it, I put a link on the bottom. Yeah, an affiliate link. Look, paramecium. Now that's going to be a challenge. Okay, so that is paramecias. Those ciliates are best observed alive. Okay, making a mound of paramecium is a challenge because these water organisms, they like to deform a little bit. Somebody actually, it seems to be better. Okay. Well, thank you for actually mentioning this. If the video was lagging, I was actually recording the video at the same time while I was streaming. Okay, and I stopped the recording now, so maybe this was the issue. So paramecium whole mount. Now I really want to know. That's going to be a challenge because making a mount of paramecium, and then usually the density also has to be right. So let's go down a little bit. I'm going to switch into DIC. Here we go. It's stained. Okay. I don't know what they used to stain it, but that is, and I have to tell you, I'm quite happy that it kind of did retain the original shape. And I wonder if those fuzzy things that you see on the outside of the cell, I wonder if these are the cilia. So we got to go up with the magnification here. Gonna go up with the, yes, they seem to be the cilia. Oh, I bumped it. Here we go. That's actually quite nice. That we're able to see the cilia. Of course, the inside we're not able to see quite, I bumped it again. The inside I did not see. We do not see quite well. Let me remove this DIC thing here. Let's close the condenser a little bit. Close the Köhler. That's too much. Okay. Good. Now 60 times. Yeah. Paramecium stained. Okay. So I'm going to explain this here again a little bit. What we're seeing here. I do have to say that these cells are. I'm 60 times now. A little, yeah. Seem to be a little bit smaller than the other ones that I have. So in any case, paramecium, that is a single-celled organism. It's a ciliate, it's a protist. It's actually one of the larger single-celled organisms. And these are so-called ciliates, called ciliates because they have cilia, and cilia, the tiny little hair on the surface. And they use these here move, and these here basically allow the paramecium, the cell, to move through water. So it's a freshwater organism, and those cilia, they beat so quickly and so fast that it's difficult to actually observe them live, because first of all they beat so quickly, and the cells, the paramecium, also they move around in a way so quickly that it's almost impossible to chase them. So what you have to do is you have to really use a very little water to compress them between the cover glass and the microscope slide so that they are not able to move. But in a permanent mount like this, where they are stained red, we can actually see them much better. And we are also able to see the tiny little hair, the cilia, which is also quite nice. As a matter of fact, this specimen is way better than the other one that I have, the other permanent mount that I have. So let's see if we're able to find a few others. Let's go down yet further with the. Look at this. This one is blue. Why is that? Did they use different? How did they do that? Could it? I don't know. Maybe. How did they stain them differently? Did they mix them somehow? Could it be that they maybe added one first and stained with one way and then added the others? I have no idea. But that's quite interesting. Yeah. But this is essentially, they have used two different stains. But when you use two different stains, then I'm kind of surprised that they're kind of next to each other. So maybe they mixed the samples. Interesting. Very nice. In any case, very nice to see the cilia again here. That's quite fascinating. I mean, for those of you who are a little bit into bacteriology, you might have heard already of gram staining, where you're also able to get so-called gram-positive and gram-negative bacteria. One of them, they stain dark blue, the other ones stain red. But that depends on the way that whether they have one or two cell walls. But for the paramecium, they're all actually the same. And so I'm a little bit surprised that there are essentially two different, that they're stained greenish, blue, and red. It's quite nice, quite interesting. Yeah, that's essentially over here. Yeah, a red and blue batch. They made a batch with red cells, a batch with blue cells. Yeah, and then they probably mixed them. Yeah, that's the only way that I can imagine this. You apply one of them. Yeah, a drop. You apply the second drop with stained differently, and you allow it to dry, and then you add the cover, the mounting medium. Yeah. Okay, video is still lagging. Interesting. Yeah. So so let's see. Let's move on. If the video is still lagging, then I hope that, yeah, maybe, yeah, I hope it's not too bad. And otherwise, you maybe have to, if some parts are missing, maybe watch them afterwards. Drosophila are fruit flies. So that here is a fruit fly, whole mount of a fruit fly. We're now moving into the insects. And this actually, that's quite nice. Look at this. Let's go down with the magnification a little bit further. Okay. So that is now Drosophila, the fruit fly. Look at this. So insects are always a little bit of a challenge because of their thick chitin and exoskeleton. That's basically the outside part of the insect is usually very dark. And therefore, usually when you put insects under the microscope as a whole mount, then you're just going to see completely dark. But here we're actually able to see that it's quite transparent. And this basically means that they have bleached the insect, or must have bleached it somehow to be able to make it brighter. Okay. So yeah, you might actually yeah, we've got a whole bunch of fruit flies also in our kitchen. So if you let some kitchen some vegetables stand around, or some fruits, yeah, then you see those tiny little flies. That's basically what they are. They are studied extensively in research, especially in developmental biology and in genetics. And they are model organisms. Yeah? So let's go up a little bit with the magnification. Let's have a look how this actually looks like. Yeah, actually, this is the head. Yeah, look at this. The compound eyes, which are normally completely dark, are now very bright. Okay, yeah, they have definitely processed this. Yeah. I also like, do they have one or... fruit flies belong to those insects that only have one fully developed pair of wings. Many other insects have 2 pairs of wings. And I just wonder where the second pair is. Maybe they actually did remove it so that they don't overlap. No, there is. It is here. The second pair is here. It's folded here on the top. Yeah. Huh. Very nice, actually. Let's go yet further up with the magnification 20 times. Yeah. Preparing a specimen like an insect is generally always also a little bit difficult because during the preparation process, it's of course squashed together, and this deforms the insect. And yeah, here we see the tiny little hair on the wings. Oh yeah. Yeah, okay. Huh. Quite nice. Okay, I'm gonna go through the questions again. Okay. Yeah. It would be nice to see live just a second, I cannot see this now. Just a second. See live insects with a compound microscope. A Drosophila on Vaseline can do it. Okay. That's an interesting one. I have to be honest with you. I tried. Look what I tried. I tried to catch some of those Drosophila flies and alive, and to kind of put them under the microscope, stereo microscope, for example. And what I tried to do, and it didn't work, but the Vaseline is a nice suggestion. You have to kind of make it sticky. You have to somehow make the surface sticky. And what I tried to do is, can you see? Yeah, I tried to use this double-sided tape. It's very strong double-sided tape. Yeah. And however, the flies were able to run over it, so they did not get stuck to it. Yeah, so they were always cleaning their little feet, I could see, but the tape was still not strong enough to actually Yeah, yeah, to actually catch it. Okay, so that was the fruit fly. What's another one? Antenna of a butterfly, hole mount. Butterfly antenna are actually have a very large surface area because they have to detect the so-called pheromones. Those pheromones are those things that. Okay, I just why can I just ask you something? Why does this not show the whole image anymore? Just a second. This is kind of interesting. Just a second. Huh. Okay. Interesting. Okay, I was kind of hoping to be able to. Why did I not stretch this? Let's stretch this all the way. Just a second. Why is the frame? Let's see if I'm able to stretch this a little bit more. I want to see the whole thing here. For whatever reason, the aspect ratio changed a little bit. This is kind of interesting. Okay. So here we go. Okay, I was just, there were these black borders on the side, which kind of surprised me a little bit. Okay, here we go. So that should be now the antenna of a butterfly. Well, that's interesting. Yeah. So when we go in, yeah, then we can also see that there are lots of tiny, I think, either scales or hair. Interesting. Never seen that before. But honestly, for the price, I'm quite overall, I'm quite happy with that. I don't know if you've seen my live stream several weeks ago, not several, even a couple of months ago. I bought myself a very cheap slide set on plastic slides. And yeah, they were indeed a little bit not so. Of course, I mean, you get what you pay for, but I think that those slides here at least are quite useful. Yeah, there is a little bit one general criticism that I have to basically almost all slide sets that I've got so far, and that is that yeah, what do you actually see? I mean, you see it on the label. What it is, but and then you look at it, but sometimes in order to understand what's going on here, yeah, it might not be very intuitive. Yeah, and that is a little bit of a pity that I think is what we actually would need is we need some kind of a booklet or manual where I can actually get some basic information about the specimen. It's not only how it was prepared, but essentially, yeah, what is the significance, the biological significance, just some basic description. And this is something that essentially I'm kind of missing a little bit. Yeah, yeah, you see a lot of cross sections of plants, and it all looks very nice, sure. But what are actually the parts that I see? What's the significance of everything? Planaria is a flatworm. I don't see. This is a very transparent specimen. Okay, it's a very transparent specimen. So I don't know if there is the. If I'm able to see a lot here. Transversal section, a cross section. Yes, okay. This is, yes, okay, got it. Yeah. So this is basically, because it's a cross section, you have to imagine the flatworm kind of extends outward of your monitor, and we just cut it across like this. So basically, if this is the flatworm, then yeah, it was cut, yeah, like this, and we're looking at it from the side. Yeah. So this basically means that when we zoom in, yeah, this is how it looks like. Okay. You can see the different layers, the epidermis on the outside. Yeah. Yeah. It must again have been stained, because we do see. But the contrast is a bit low, I just realized. Okay. Yeah, again, if you look at this, for example, yeah, and yeah, okay, what do I actually see? And a short description, especially for beginners and so on, would be quite nice. I can make sense of the whole thing is because I know, okay, on the outside you've got the epidermis, it's the outside layer of skin and so on. But yeah, the dark dots are possibly nuclei of the cells stained. Yeah, here on the top we can see now the different cell layers. But it would be kind of interesting to have a little more information, even not about the specimen itself, but maybe about, okay, what are flatworms? Okay, where can you find them? What about this specific one over here? Yeah, it must have been from a certain species because they've been breeding those. Probably, yeah, I would say maybe a planarian. Yeah, so this is something that would be kind of nice. I mean, I already have. I'm going to float an idea. Okay, and maybe some of you, maybe sooner or later, some slide manufacturers are listening. I don't know, but it would be kind of nice to have a little QR code on the slide, and then when you take a picture using your mobile phone of the QR code, then you can be connected maybe to a website. Excuse me. You could be connected to a website where you can get more information here. Huh? So that's a planarian. TS for transversal section. Huh? So desk again. So yeah, okay. Let me go quickly here through the comments again. Yeah. Okay, some people still seem to have issues. I don't. It's a real pity. Yeah. So let's move on here. Male worm and female worm, which is kind of interesting because many worms actually are hermaphrodites. And yeah, sure, male worm. Which worm? I have no idea. Huh? See, and that is something that I'm kind of missing a little bit. And how can it be? It's a male worm, really? Yeah. Here we go. Also, yeah, again, the tissues are very thin. Or maybe it's a cross-section through the male gonads, the male reproductive organs, because many worms are actually both male and female at the same time. Yeah, see, looks all very nice. But then again, I'd like to know a little bit more. Yeah. So it would be interesting to have a little bit somehow a description somewhere. So if there would be a QR code, maybe with a picture and then a labeled picture explaining the different parts here. This could be maybe, if this is the cross section, this could be maybe the how do you say, the digestive system on the inside. So let me remove this here. This is how the worm looks in complete cross section here. This slide here is a little bit dirtier than the others. You see there is quite a bit of stuff on here. But still, yeah, relatively still okay, I would say. Yeah, at least the specimen is flat. Yeah, on here. Okay. Yeah. Okay. Let's move on. So it then basically says female worm, whatever that means. Female worm. I'm kind of wondering. Oh yeah. Somehow maybe these are the places where the eggs are stored. I don't know. Oh yeah, but this one actually does look better than the other one. Looks much more contrasty. Yeah, actually significantly better. Yeah, it looks like it could be that the cross section is also thicker. Quite quite fascinating to look at, but I wonder, yeah, what these parts all are. Possibly, I don't know, places where eggs are made, I don't know. Let's have a look here further. Okay. Yeah, there is already a comment in just instead of just selling a box of slides, they should include a drawing with key features. Yeah, exactly. And this is indeed something that I would say would be really nice. You know what? I mean, honestly, yeah. Then I'm already thinking if I should not do something like that. Yeah. But this looks patterns of nature. Yeah, another thing that I want to mention here in this context, we're looking at animal tissue right now, and you see that it looks quite remarkably different from plant tissue, because in plants the cell walls were so easily visible, and here, of course, the cells are not quite as easily distinguishable, because they are only separated by a cell membrane, which is very thin. And this is also a reason why it took a little bit of time to devise the cell theory that all living things are made of cells. To my knowledge, it was actually Matthias Schleiden who first proposed that all plants are made of cells back in the 19th century. And that's, of course, it's easier to say that all plants are made of cells because the cells are more easily visible because of the thick cell walls. In animals, of course, you can see it's not so evident to see the individual cells. Yeah. So so I'm going to go through. Yeah. A question here: if you buy a prepared specimen at LIDA, will you get any description added to each specimen? Let's put it this way. I mean, we do have in school a lot of specimens from the company LIDA. However, these are somehow already a little bit older. The thing is that you have to check the catalog. If you go to LIDA, they do have a PDF, and they do have some teaching material as well. But to my knowledge, it's not like this that they provide specifically a separate description for each slide. Okay, at least this is not documented in their catalog. But I know that they do have teaching material as well, which is a little bit more generic, but I think it's not slide specific. You'd have to check, but at least I was not able to find that. Yeah. Is this rainform sized or smaller? Let's put it this way. I'm going to go all the way down with the magnification. And it's now 4 times. And this is now the complete cross section of the worm. I don't know if it's a rainworm, an earthworm. I suppose it is, but we can estimate the diameter of the worm by looking at it, because you can actually see it. Yeah, it's about 5 millimeters across. You see this thing? Yeah. So that's about the diameter. Yeah. So again, also the label here. Yeah, I'm not so happy with that. It's cross section of a worm. Which worm? Okay. So mouth part of a honeybee, Daphnia, Cyclops and Daphnia, earthworm. Well, there is a separate earthworm one here as well. 58. That's interesting. Okay, see, this one here, earthworm. Cross section. It's just like just like the other one. Where is the microscope slide? Here we go. Yeah, you see that this one is again a little bit thinner. Okay. Yeah, so it looks everything looks more pale. So let's go back to the insects. What is this here? No, it's not an insect, but that's a cyclops. That's a water crustacean. And let's find it. Yeah, here it is. Also stained. Yeah. Honestly, they look better alive. But alive, they're more difficult to observe as well because they move so quickly. This guy is a little bit crushed, of course, because they're kind of thick and therefore but yeah, some features can be seen more easily because simply the animal is not moving anymore. I don't know if it would have been necessary to actually stain it, because it's all of it is red. Yeah? But I can imagine that, especially if you're doing microscopy, if children are using this, or for example, people who who are just learning microscopy, like in school, yeah, then having a strongly stained specimen makes it sometimes easier for them to find it. Yeah? Daphnia, okay, that is another water flea. Okay, here we go. Yeah, this guy was also a little bit crushed. Yeah, this guy's pretty badly crushed. Yeah. Yeah. And maybe, I wonder if these round things on the back here are some kind of eggs or something. Yeah. The eye you can see. So if you want to observe live Daphnia, then I recommend that you just put them in on a slide without very little water, but without a cover glass, so that they're still able to move around freely, but because there is so little water there, they're not able to swim away so quickly. So the surface tension kind of holds them in place. Yeah, but yeah. Of course, when you're preparing permanently mounted slides, then you have to apply some kind of a mounting medium. You have to apply a cover glass, and this, of course, crushes them. Okay. So what else do we have here? Here, the mouth parts of a honeybee. Oh, this seems to be, look, I gotta show this to you here on the desk view. Look, seems to be, is a very well visible, so it seems to be a very dark, darkly stained, dark specimen. So, here we go. Oh yeah. So that is basically the mouth part of a honeybee. Okay. This actually also looks quite okay. Okay. And yeah, here it's becoming quite dark. Let's open it up a little bit. Okay. Yep. Very dark here. But still, I would say it's fine. Let's go down with the magnification here. This is now, yeah, the mouth part. That's basically where they're collecting nectar. Okay. So, what else do we have here? Sure, you should start a business of slides with explanation schematics, selling them to school. And do you, yeah, honestly, I've been really thinking about this. About actually making slides with, or I don't know, buying a whole bunch of slides and then slapping some descriptions on it. I don't know, yeah, or a booklet. That's certainly something that would be possible. That's time consuming though. Yeah. And when you're buying prepared slides from a company, then of course you don't always know if you're getting exactly the same things. So that is now a housefly. So the one before was a honeybee, and that is now a fly. Okay. The front part is missing here. Okay, here we go. Yep. You can see all of the hair. Yeah. Yep, I'm going a little bit faster. So what else do we, oh, there's still so much stuff. Oyster gill, ant, whole mount, 87. Yeah, they've got quite a few of these things here. The good thing here is that this slide box does contain quite a few specimens that people can identify with. Yeah, everyone knows what an ant is and a honeybee is and so on. Yeah, so these are animals and everyday kind of, yeah, that. Ooh, look at this. An ant and look, two ant heads. Yeah, some body parts missing. But at least you can see those, yeah, the mandibles. Yep, that's the head of an ant, and that is the head of an ant. Antenna is even present here. Okay. Yep, some of the legs are a little bit cut off or missing. The abdomen popped open. Now, what could be is the following. When you prepare insects, what you do, because if you want to make sure that the insect is, that you can make a proper mount, you have to actually make sure that the contents of the insect, so to say, is removed. Otherwise it starts to decompose. And what you do is you put the insect for some time into potassium hydroxide, KOH, and the potassium hydroxide will bleach the insect. And then you can, and it will make the exoskeleton very, not so brittle anymore. And then when you compress the insect and the exoskeleton, like here, it will pop open. The contents can be removed, and this basically makes it possible so that you can make the insect more flat. Yeah. So an ant, if you were to put an ant directly under the microscope like this, then it's just going to be completely black. Yeah. But I'm quite. I think they might have treated this insect just like the others with some potassium hydroxide. That's actually a very common way of preparing insects. Yeah, yeah. Double-headed, yes. Yeah. I would like to know which ant is that. I mean, it's kind of. I mean. Don't want to complain about this, but I mean, these folks are doing this as a business, yeah, and it would be nice simply to have a Latin name on here, or just a name of which ant it is. Yeah, yeah. So can you use DIC? Currently, I'm just using regular bright field. So let's use. I'm just going to shoot DIC, but sometimes. You don't get for some specimens, you don't get a very good effect, DIC effect, because especially for the thick ones like this one is now DIC, yeah. But you see, I'm changing the prism here, not a huge difference. Yeah? So this depends quite a bit on the specimen if you actually get a good effect or not. Yeah. What concentration of potassium hydroxide do you treat the insect and how long? I don't know that for right now. I have not done it myself, but I have basically, when I've done insects, I mounted them directly in U-Pearl mounting medium. So I put them into alcohol and put them into U-Pearl, and this also bleached them. But there is a video that I've found online of someone who is doing this quite extensively. So you can actually try to find that video. I don't know the concentration that is used, but it's probably not going to be so low. And somehow the base reacts with the pigment, I would say, in the exoskeleton, and then this bleaches it, and it makes it also quite soft. Yeah? And then what you have to do is you have to compress the insect between two slides and put it into alcohol to remove the water again. Yeah? It's not such a complex procedure, yeah, and I've read in some books that essentially they did not specify the concentration either. So I would probably suppose that this depends also on the time that you want to use it. If you use a very concentrated KOH solution, then maybe it's going to be much shorter. Aphid, yeah, those aphids they basically suck the sap of plants. If I find it, here it is. Okay. This one is actually much nicer again. Look, this leg part missing here, but it's okay. This one actually looks kind of complete otherwise. Okay. Yeah, so there are again people who are having streaming issues. I don't know why this is the case. Yeah. This seems to be some kind of a network issue. I normally did not have those problems before. But what I will try to do for next time is I'm going to go down a little bit with the bit rate. Maybe this improves something. I don't know. So yeah, aphids. These are those little critters that have been a little bit tough also on my plants because they have been sucking the sap quite a bit. Yeah, frog blood smear 97. That's gonna be the one where we hopefully are able to see some of the nuclei. Just a second. What is this here? Frog blood smear. Okay, so let's see. Amphibian blood contains a nucleus. Yes. Ah, look at this. It's been yeah. They've somehow put the blood into some kind of a matrix. And what we see over here is, go in here. Yeah, yes, we are able to see it. Let's go up further. These are the red blood cells of a frog. And there is something very typical about those. How they are different from, for example, human red blood cells. Okay. Notice that those red blood cells have a dark spot in the center. That's the nucleus. Human red blood cells do not have a nucleus. So during the formation of the red blood cells in humans, in mammals, the nucleus is an asymmetric division, so without a nucleus, because the space is needed to carry hemoglobin. And amphibian blood, of course, frogs are amphibians, they have a nucleus, and that's the dark red dot that you see in the center. And this is a piece of information that I know, but which is, of course, missing on the slide. Yeah. So that would be something that would increase the value of a slide set like this enormously to have some kind of a manual. Yeah. So there is a technical question. Is it specifically made for fluorescence? No, the FL, if you see the question over here, is this FL label here? Yeah. FL stands for fluorite, not fluorescence, it's for fluorite. And those fluoride objectives are semi-apochromatic objectives. Yeah. Can be used. Yeah, so basically a fluoride objective are not quite as expensive as the apochromatic objectives. Yeah. So can it be used for fluorescent microscopy? Well, I think that's a different issue, because in fluorescent microscopy what you have is you sometimes illuminate the specimen also from the top. So this basically means that there can be objectives that are specifically made for fluorescent microscopy, but they do not have to be fluoride objectives. Let's put it this way. Yeah. So the FL is just coincidence that this means it sounds similar to fluorescence. Yeah. I'm using an Olympus microscope. An Olympus microscope with a camera attached, and which is quite nice. I got this microscope because I'm doing, of course, lots of live streams and so on. It's this is also a microscope which is used quite a bit by doctors. So when I bought the microscope, I was told it's one of the microscopes that pathologists, doctors like to use a lot as well. Yeah. Brand is not so much the issue, but yeah. Over the skin of a fish blood smear, fish gill, aphid ants. What else do we have? Euglena 49. Euglena is another single-celled one. So let's have a look at this. It's also something that I otherwise don't have. So let's see. Lots of them. Very nice as well. Euglena are also protists, and they can do photosynthesis. So that's actually one of those, how do you say, single-celled organisms where there's a debate, should we count them more to the plants or are they not plants because they can do photosynthesis as well? No, they're their own category. Yeah. So again, it seems that they used 2 different stains and then combined the batch, and yeah. Euglena has a flagellum, which is a whip-like extension, which is of course not so easily visible right now. Yeah, 60 times. Yeah. Because it's very thin. Yeah. Yep. Okay. So let's go on. What time is it? Yeah, wow, I've been already doing this for over an hour. So maybe I'm just going to do this for a couple of more minutes here. Let me quickly look through here. This is a pine cone. This is seems to be also quite nice. It's a pretty large specimen. And yes. Okay. Yep, that is very similar to the other slide that I have. That's a pine cone cross section. That is a female pine cone. I can tell you that because these are the ovaries which will develop into the seeds, and I know that because I've got another slide. So that's a female pine cone. Yeah. So that's one of those ovaries which will form into a seed. This one maybe possibly as well. Yeah. So, but let's go down with the magnification so that you can get a better overview of how this whole thing looks like. Yep. Yeah. It's a fairly large specimen. It's approximately 1 centimeter or maybe even longer. Looks quite okay, yeah. So nothing is folded back onto itself. Yeah. Again, I would wish that they would cut it a tiny bit thicker. Yeah, then it would be a little bit more stable, and then you do not have those ripping and tearing of the specimen, and the whole specimen also will get a little bit more volume this way and will become a little bit more contrasty. But I think it's okay. Yeah. The one slide that I have from Lida with the same one is a little bit thicker and it looks a little bit better subjectively, yeah? Because there are not so many wide empty spaces. Yeah? But each aspect has its advantage and disadvantage. Okay, so I'm going to check here again. Okay, this is an interesting one here. Would be interested to see a specimen related to various pathology. So basically, normal versus pathological tissue. Basically the thing is the following: there are indeed microscope slides out there that can be bought, which does contain tissue, for example, from cancer biopsies and so on. So these things can be bought. They are quite a bit more expensive, obviously, because I mean these slide sets here, these are just standard slides of I would say very common organisms, right? But if you want to buy very specific slides, I'm just gonna type it again into the messages here. You can visit the follow. I'm not affiliated with them, okay? So I'm not working for them, and yeah. But there is a company, and you visit the web page. I type it in right now, lida.de, and you can download. You can download a PDF catalog from them, also in English, for free. You just download it, and they contain all slides, whatever. If they don't have it, no one has it, right? I just have to. There is a separate price catalog that you can download. I just want to warn you a little bit. They are a little bit more expensive, actually quite a bit more expensive than those slide boxes here. However, they have highly specific slides as well. I think the quality is, I've got in school, we are using slides from that company, and I would say that the quality is really good from them, right? They say that all of the slides are made in Germany. Okay, so they have specific laboratories, and they also have, they say that some slides are, if you need of very rare specimens, you have a waiting time because they will actually make the slide for you. So this is simply, simply a recommendation. Again, I'm not affiliated with them or anything. Yeah. Do you have a specific lab where you perform the just a second, I cannot read this because this microscopic activity. Okay, I've got a separate room, and basically I've got a slightly larger table where I've got on my left and on the right side, I've got the microscope in front of me. I've got the monitor with camera. You know what I'm gonna do? See, that's the monitor. I've got 2 webcams on the top. Yeah, that's my desk. Okay, the microphone is here. I've got over here the stereo microscope, and on the window, yeah, I've got a couple of water samples. Yeah, it's basically how it looks like. Yeah, if you, I do also have a second YouTube channel called Microbehunter Microscopy. There's a link somewhere where I'm basically just answering some technical microscopy questions, and this is a little lab that I have arranged, a little studio, so to say, that I've arranged in the cellar, right? Yeah. So concerning another thing is that there are also, the company also, Leader also sells slides I've seen with basically pathogens. So for example, if you want to see bacteria that causes tuberculosis or the bubonic plague and so on, they even have slides with those bacteria as well. So this is and Conus. There's a comment here that Conus apparently also sells slides, right? Yeah. So that is basically, the thing is, shall we look at another one as the last one? Locust wings, mosquito wings, honeybee antenna, fly antenna, pollen germination, number 36. Let's try that, because I tried to lily pollen, just a second, pollen germination. So I wonder if you're able to see the pollen tubes. Yeah. So that is pollen. I don't know which kind. See? This is a little bit. Yeah. Yeah, let's have a look. Again, stained differently. And let's go in yet further. Yep. Yeah, so this is pollen of some plant, obviously. But what I'm not able to see is I'm not able to see the pollen tubes. This is actually, when they say germination, then this is actually something I would expect. Because when you add some sugar water to pollen, and if the conditions are right, what's going to happen is that you're going to see that there is a little tube starting to grow out of the pollen grain. And that tube is called the pollen tube. Yeah? And this is basically what starts to grow when the pollen lands on a plant, which is compatible. And then this pollen tube will start to grow. And you can also trigger the growth of the pollen tube by adding some sugar water. Not all pollen do like to germinate then, but here I do not see any germinating pollen. Doesn't matter, it looks nice. Yeah. But this was a little bit my hope, because I could not always get the right conditions for germination. I've tried this during springtime several months ago. I've tried it to do this again. And some of the pollen germinated, some didn't, and the conditions were not always quite right. Yeah? So I'm still experimenting around here. So let me see. Yeah, fern leaf, no, sunflower. We looked at this. Okay, another one. Housefly, butterfly wing. Butterfly wing 77, and let's have a look at this. I expect to see some of the scales. The good thing about this is that the slides are actually numbered. Butterfly wings ocalis. Okay, let's have a look. Ah, yeah, here we go. And this looks fine. Yeah. As expected. Yeah. So if you are observing sometimes some water samples or dust samples, I cannot believe this. No. Oh my gosh. Okay, I gotta explain this. Do you see this weird structure here? This weird long structure? Okay. This with this very thin extension. It's the second one here. Many years ago, I found them in a dust sample this year. And I was wondering, what in the world is that? Okay, what in the world is that? Yeah, and I had no idea, and I kept on finding it again. And then I posted, this was 10 years ago, maybe longer. I posted a question in the forum and with a picture, and then somebody actually says, yeah, these are the hair of a so-called a carpet beetle, of the larva of a carpet beetle. And those carpet beetles, they basically, if you have them, they like to eat carpets, obviously. And this was basically one of the so-called the setae. The setae are the hair of a carpet beetle. And then I actually did some searching again in the dust, and I found some dead carpet beetles indeed, and they had thousands of these structures. And here, right now, on a butterfly, on the scale of a butterfly, we're seeing, yeah, they go kind of crisscross, like, I don't know, this seems to be some kind of contaminant maybe. Yeah, but that is really interesting. Here again on the top. Yeah. It's I call this the mystery object. Yeah, and you know what? This is really interesting. Yeah, yeah. So this is yeah. Yeah, these exactly. I mean, you know what I'm gonna do is I don't have time right now, but maybe that's actually the topic of next live stream, or I'm gonna make a video, and I'm going to talk about this because I've actually found quite a few of these here in the dust, and here quite surprised that on the wing of a butterfly, yeah, they're also here. I don't know why. Yeah. Here on top as well. Interesting, fascinating. Didn't expect this. So, yeah. So this was really a surprise. Yeah. So what I'm gonna do now, people. I hope that I didn't overlook any of the questions here. Let me quickly go through the remaining comments. Yeah, you answered all of the questions. Very nice. What a wonderful hobby. I think so as well. Okay. Carpet worms. Yeah, so these carpet beetles, you can actually research them also in Wikipedia. It's not the beetle itself, it's the larva of the beetle. So basically it's like a little caterpillar, and it's totally hairy and furry. And if you look at it under the microscope, then you're actually going to see that those hair are those structures that you see here. Yeah, yeah. So yeah, so what I'm gonna do now is I think I'm just going to call it quits for today again. Yeah. So hope that you liked this. This is as always a completely improvised session, because it's also the first time for me that I'm looking at those slides here. Okay. Yeah, and there is the issue that I'm going to look into that essentially was a little bit lagging. What I will do is, I will try to reduce the bit rate a little bit. It did work last week, I don't know why, but I will try to do that, and then maybe next week I hope it's going to be a little bit more fluid and with less interruptions, and maybe you can next week give me some feedback if the image quality is still fine. So when will it? Just before I go on, there's a question: when will the next live stream be held? I always plan to do the live stream on a Saturday at the same time as you were watching right now, which is 9:30 in the evening Central European time. I will usually several hours in advance always put the live stream online so that you see this countdown timer. If for whatever reason I'm not able to do the live stream on that time because maybe there is some I don't know, I have an appointment or so it doesn't work out with my schedule, then I will put an announcement. I will put an announcement in the community forum of this YouTube channel, and then I might move it maybe to Sunday, 24 hours later. Okay, so this is usually the thing. So do keep on checking for this announcement, or you just go on my homepage microbehunter.com, and I always post also embed the YouTube video as well, so you can also find it easily there. But usually a couple of hours in advance, I will always put this trailer so that you know what the video is going to be about. Okay, I'm going to call it quits for today. Have a nice week. Check my videos on the YouTube channel as well, if you're interested. Also the YouTube shorts that I'm publishing every now and then. I wish you all the best. Happy microbe hunting as always, and see you around next time. Bye-bye.